Other than that, I haven't grabbed it. I like going through those, and then it's like I gotta like go through and like get things.
Yeah, but like if you so I guess
Okay, welcome back. So, we are on the supergroup Archaeoplastica, which are everything. So the thing that these have in common is that there was a primary and symbiosis in that thing, right? So everything here swallowed a cyanobacterium, didn't digest it. Okay? All of the other photosynthetic protists, of which there are many other groups, swallowed one of these in turn, with the exception of that really weird other primary endosymbiotic event that we talked about among the circozoans last time. All right, so we have already talked about red algae. We'll talk about green algae today. And actually, specifically, we'll focus mostly on the Clora Fights. The Charophytes we'll talk a little bit more about when we talk about land plants, because they are the sister taxon to the land plants. Okay, so we call both of these things green algae, but we don't call this green algae. What's the word for this when we do that? Are these plants? Are chlorophytes and charophytes plants?
they are paraphyletic.
Yes, exactly. So this is a paraphyly. Because, well,
Because they have a pair of common ancestor, but doesn't include
one of
plants,
the,
which is another descendant.
exactly. So it's a group that includes everything and the ancestor, but doesn't include some of the extant lineages, namely the plants. So we don't call plants algae, right? Even though we kind of technically should. Let's do it.
Write that letter.
Dear science community. Okay, so plants are descended from green algae, but we don't call them. So among the green algae, right, we have these two distinct lineages, one of which is more closely related to plants than either is to the other lineage of green algae. Okay, so charophytes we'll talk about next time. Chlorophytes, let's talk a little bit about those today. Okay. So most chlorophytes, right, live in freshwater, but there are several marine species as well. So if you see something that like sea lettuce, right, that's an example of a chlorophyte. So chlorophytes, there are single-celled chlorophytes, so single-celled green algae. There are multicellular green algae like the sea lettuce. And then we get these guys, the Cholera, I don't know how to pronounce that, but that's a single-celled organism. What? Yeah. So, yeah, there aren't multiple cells in this structure. Look at the scale of that.
It's a whole cell.
It's a whole cell.
That's one cell.
That's pretty wild. Yep.
What?
Yeah.
So what's like the
Does not
biggest
compute.
single cell like in the
I mean,
would that be it? Or?
so are we including like Plasmodial slime molds as a single cell because technically they're not separated by cell membranes. So it's a tough question to answer, right? Because some things, like during different stages of their life cycle, just become one giant blob of many nuclei, right? And there are actually cells in your body that are syncytial like that just merge. So you have like one giant cell with lots of different nuclei in it. In terms of cells with a single nucleus, I don't know, but I imagine that this guy might be in the running. Okay, you also find chlorophytes as the symbionts in some lichens, right? So we talked about lichens, I'm sure, in the course, yeah. Just as a refresher, a lichen. See if there are any within range of where we can see.
Let's go on an adventure.
Okay, yeah, here we go. So we've got some, see that tree with all the like crap growing on it? Like right across the walkway there, the tall one?
Yeah, those pale spots.
Yeah, exactly. So you might think those are mosses, and some of them might be mosses, but the other stuff that's not mosses are lichens. And lichens are a symbiotic community of organisms that we just call lichen. We're just like, you're actually many different things, but we'll just call that whole like superorganism a lichen. So lichens usually consist of some kind of fungus, some kind of photosynthetic algae of some sort, many of which are chlorophytes, and then often also bacteria, occasionally cyanobacteria. Okay, so the fungus provides like a stable structure, a nice microenvironment for the algae to grow in. So that's what they give to the relationship. And the algae photosynthesize, so they're fixing carbon, making sugars that they give to the fungus, right? And then the bacteria, that's actually a pretty recent addition, like we're talking within the past decade or two. They're realizing lichens are also bacteria. And a lot of them are nitrogen fixers. So they're taking atmospheric nitrogen and converting it into organic nitrates and nitrites that can be accessible to both the fungus and the algae. So it's just like this really cool sort of microcosmic community of all of these symbiotic mutualistic relationships. So anyway, chlorophytes are one of those mutualists. Oh, okay, I'm sorry. So Cholera actually do have many nuclei, there's just no cytoplasm. So it's very similar to Plasmodial slime molds. Okay, so there are many different ways to get a large-sized organism, and chlorophytes do all of them. They form colonies of individual cells. Someone want to fact check me, are Volvox chlorophytes? I believe they are, but I want to make sure that's true.
Chlorophyte green algae.
Sweet. Okay, so Volvox are a perfect example of colonial chlorophytes. Then we've got true multicellular bodies where there's cell differentiation, so there's cells that specialize in doing different things, like we saw with the sea lettuce. And then we've also got repeated cell divisions with no cytoplasmic division. So there's no cytokinesis, there's just nuclear division, like we saw with the Cholera. Okay. I believe you've already seen this picture. Okay, a lot of chlorophytes have really complex life cycles where they alternate between asexual reproduction and sexual reproduction. So let's take a look at one of those life cycles. This is a life cycle of a Chlamydomonas, that is the genus. And you guys will see, I think, some of these in fixed slides on the microscope today. So they're unicellular chlorophytes, and they can go several bouts of asexual reproduction. And there's two really weird things to note about these haploid zoospores. First, they're biflagellated, so they've got two flagella, and also weirdly they have like a U-shaped chloroplast. So you can get two of the mature zoospores fusing with one another, and you get fertilization. When they are fertilized, they form a zygote, and that zygote is super hardy. It's like very resistant to all kinds of things, which I did not note. Okay, but I'm sure it's desiccation, like extreme weather conditions, UV radiation, right? So if you're going to go through like an extreme period of not good stuff, it's probably a good idea to have sexual reproduction and just like hold tight as a zygote for a while until conditions get better. When conditions get better, undergo meiosis again and release those zoospores. And actually, this is a really common pattern in organisms that can reproduce both asexually and sexually. They have asex while like the getting is good, right? So even aphids do that. They'll have asexual reproduction until your garden is destroyed. And then they're like, there's nothing left. We have to move on. Then they start having sexual reproduction, and that's usually like the dispersal time. So any aphids that can fly, they're the ones who are sexually mature, and they can go on and have sex. So fun fact. We're not on invertebrates, but this is a really common pattern where you basically have sex to escape something that's gone bad. There's a joke in there. I'm not going to go for it. Okay. All right. So we've talked about the Archaeoplastida. Let's talk about Eunuchons now. So the fourth supergroup is Eunuch Canta. And these are the protists that are most closely related to fungi, and they also contain the protists that are most closely related to animals. Okay, so technically this group Eunuch Canta includes both fungi and animals. We just don't call them protists. Okay, we can largely divide these into two groups, Amoeba Zones and Opisthocons. And Opisthocons contain animals, fungi, and their close protist relatives. So I forgot who it was, but someone looking back at that original phylogenetic tree with the 4 supergroups of eukarya noted that it looked like a polytomy, right? Someone said that. And the reason is because they're not quite sure like what the relationships are between those 4 supergroups, but the current dominant hypothesis is that Eunuchons are the ones that are most distantly related to all of the other supergroups. Okay. So these groups form the Amoeba Zones. So Amoeba Zones include slime molds, tubulinids, and Nth Amoebas. And these groups form the Opisthocons. Okay, so it's including fungi and their closest protist lineage, sister taxon, the Nuclearians, and then animals and their closest protist lineage, the Choanoflagellates. Okay, we are not going to talk about these 2 today. Okay, so we're actually only going to talk about the Amoeba Zones today, those three lineages. Don't worry too much about this slide. I almost took it out. But just suffice it to say that there is they have at least one line of evidence supporting this hypothesis that the Eunuchs are the outgroup to all the other supergroups. And the reason is because there are these two genes that get fused in all of these lineages that are actually separate genes in the Eunuchs. So they're still homologs, right? They're still evolutionarily related to one another, but there was a clear gene fusion event. Do you guys know what I mean by a fusion? Like literally the two open reading frames of those genes just get smushed together.
Like the stop codon.
Yeah, imagine, there's a stop codon that just vanishes between them. Yeah. So geneticists can engineer fusion genes at will. And I say at will, like within a couple of years of hard work, right? But that's something we do as well.
Does that make like spider silk out of like those?
Not necessarily. So in those cases, they could actually just insert complete spider genes, right? But let's say they wanted spider silk protein that also had some other protein feature. They could steal that protein feature from another gene and just tack it on, right? And then it may or may not work as expected, right? Given that it would change the shape of that final protein product. Does that make sense?
Yes.
But yeah, that is something that we can do and play with and experiment with. Okay, let's talk about Amoeba Zones. Okay, Amoeba Zones. So the word amoeba is like the word algae. We use it to describe lots of things. Not everything that we call an amoeba is even in this amoeba zone group because people are silly, right? But oftentimes, right, this is a good bet. Like if your life depended on it and like at gunpoint someone was like, you have to identify this thing under the microscope and it looked amoeba-like, a good guess would be it's in Amoebazoa, right? It's one of those three. It's either a slime mold, tubulinid, or an Nth Amoeba. Oh, there it is right there. So remember, we've actually already seen things that have pseudopodia that are thread-like. Does anybody remember the lineage that had thread-like pseudopodia? Yeah. Rhizarians. Does anybody remember one of the groups, one of the clades inside the Rhizarian supergroup? Or, sorry, the... And? Yes. Okay, let's talk about the slime molds first. Okay, yeah, so everything that, most things among the amoeba zones look amoeba-like, but not everything. So it goes both ways. There are amoeba-looking things that are not amoeba zones, and there are amoeba zones that don't look amoeba-like. So the reason these are all grouped together, it's mostly molecular evidence. So it's mostly DNA evidence.
So when you say look like an amoeba, you mean they look like they have a tube-shaped pseudopoda?
Yeah.
Okay.
Yeah.
So that's not like a total characteristic of the whole group.
Exactly. So question was, when I say amoeba-like, do I mean things that have tube-shaped pseudopodia? And the answer was yes. And then the follow-up question for those of you watching at home was, does that mean that we can't use that kind of character state as a synapomorphy? And the answer is yes, we can't. Correct, we cannot do that. slime molds are so cool, okay? Sometimes people refer to them as Myceta zoans. So anytime you see mycete, that prefix, the MYC, think fungus, right? These are clearly not fungi, right? But they are very fungus-like. And actually for a long time were mistakenly placed in the kingdom fungi. Right? But once we were able to sequence them, we realized, oh, they're actually much more closely related to all these other different protists. Not fungi at all. Okay, so we've got two lineages within the slime molds. We've got plasmodial slime molds and cellular slime molds. Let's talk about plasmodial slime molds. First, we're going to see a life cycle here. I'll walk you through the life cycle bit by bit. This is what a plasmodial slime mold looks like during some part of its life cycle, but not all parts of its life cycle. So if you're ever out in the forest and you see weird stuff like that on the log of a tree, that's what you're looking at, right? Somebody didn't come and like throw up on the log, right? That's a slime mold.
What do they feed on?
Yeah, good question. So a lot of these, right, the reason that they look like fungi is because they also basically consume things in a similar way to the way fungi consume them. So this is Physarum, is the genus. And these guys are a really common research organism. They're a model organism. And in lab, people just use oats. That's what they feed them with. We'll watch a video of it. I think I linked it. Okay, so during at some stage in their life cycle, they'll actually aggregate together, right? So they're individual distinct cells. They'll aggregate together, and the cell membranes will merge, right? So they'll actually become one giant supercell with lots of nuclei. Okay, and that's called a plasmodium. And that plasmodium actually, in times of scarcity, once again, it'll form these little stops with the sporangium on top, and inside that sporangium are a bunch of spores. Okay, those spores undergo meiosis. And then they get released, right? So they're haploid spores at this point. They get released. And you have two different cell types. You've got flagellated cells and amoeboid cells. And those can unite and fertilize, forming a zygote, which can then fuse with other zygotes to form a plasmodium.
So that picture that we have on the slide with like the yellow thing on the log, that's it in its mature preparing form.
Yeah. So as the plasmodium is when it's going out and foraging for food, and it forages for food as one unit, right? As one. Yes. Yeah, it's pretty cool, and actually on a pretty quick time scale. So people are actually starting to use this to solve interesting biomimicry problems. For example, someone just decided, I think it was an artist actually, decided to, and thanks, heads up to Spencer, who actually told me about this. So there was an artist who thought it would be cute to like basically reproduce a map of all of the stops in the Tokyo subway system, and just put an oat wherever any of the stops were, and then just like dropped Plasmodium onto this thing, right, this platform that had all of those oats, and watch the Plasmodium like basically create a network between all of these different oat sites, and it reproduced almost exactly the network of tunnels that you see in the actual Tokyo subway system, right? So it's this example of like Plasmodium basically solving the traveling salesman problem in the same way that it took humans a really long time to solve the traveling salesman problem in Tokyo, right? So maybe we should take comfort in the fact that we did it the same way that the Physarum did it. But you can imagine, right, this might be a cool way to design future, more efficient subway systems. Exactly. So there are all the stops. And I don't know what, I mean, this is clearly like a, I don't want to say stop motion, but do you guys know what I'm talking about? Time lapse, thank you. But I don't know what the time scale is. It's pretty quick. Right, so it's just optimizing like the nutrient transmission between these different. So you see it like it throws out these little threads to kind of explore parameter space, very similar to the way mycelium works. Yeah, and exactly. When it yields good results, those paths get strengthened. Here's another one where it's solving a maze. There's actually several solutions to this maze, and you'll see that it actually enriches for the best solution, the fastest solution. So, didn't mean to show this video, but it went right through it. So. All right, so over time you're going to see this pathway get reinforced the best.
Is that like cheese or something?
I think these are oats or something, some kind of grain. Pretty cool.
So this is in its multinuclear form, right?
Yes, this is so it's called a plasmodium when it's in this state where it's got lots of nuclei. Okay. All right, so contrast this with cellular slime molds. So cellular slime molds aggregate the same way that plasmodial slime molds do, but they keep their cell membranes separate. Okay. So they basically can locomote and navigate faster as an aggregate. And actually the aggregate in some species is called a slug. Right? It's not a slug. Right? It's just a bunch of cells that behave and look very slug-like. Here's a picture of that. That's an aggregate of cells. So you can see that the aggregate, right, each cell maintains its identity as a cell, right? Each cell membrane has one nucleus in it. Okay, and they do the same thing, right? You form these stalks with these little spore capsules on the top. But here is the really interesting problem with cellular slime molds, is that it's not the case that all the nuclei end up in the spore. The spore is the thing that gets dispersed, right? So basically anybody who ends up in the stalk dies. Okay? They don't pass on their genes to the next generation. Okay? So basically we have like a social organism where some individuals have to sacrifice themselves for others to survive, which is a source of tremendous conflict, right? You can imagine if there's a mutation that makes one of these guys selfish and it's just like, sorry, I have to end up in the spore. Like, excuse me, pardon me, coming through, right? What's to stop that mutation from just increasing in frequency, sweeping through the population, making everybody scramble to the top and nobody forms the stalk, right? It seems like this really impossible question of social evolution. Like, how do you get cooperation? How do you get altruistic behavior among species? Right? Same thing with ants, right? Most worker ants are obligately sterile. If you had a mutant that evolved that didn't get sterile, what would stop that mutant from increasing in frequency, right? So think about this as like a selfish mutants should invade. That's the way I always think about it, is like selfish mutants should invade these populations. And given the fact that they don't, or so it seems, what is possibly going on here? Okay, so this is Dictostelium, which is one genus of these cellular slime molds, and again, another really common model organism. So people study a lot of social evolution among Dictostelium. And okay, so there was a lot of theory about social evolution before people even were studying Dictostelium. And one of the ideas was that one way that you can get around selfish mutants invading is by having what's called a green beard gene. And the reason it's called a green beard is because in this thought experiment, they were just like, what if there's a gene that confers a phenotype, let's say a green beard, okay, so that everybody with a green beard could recognize everybody else with a green beard. And that gene also had another function, the same gene, that made those people behave favorably towards other people with green beards. Okay, so these green beard alleles were just a made-up theoretical thing, and they were just like, that's the way you would get these altruistic behaviors, is if people can recognize other altruists, which seems very not altruistic, right? They can recognize other individuals that share this trait with them and favor those individuals. So it turns out that the way these guys aggregate, like the proteins that cause them to stick together, those are green beard allele. So it basically prevents other individuals from being selfish because they have to have this protein that causes that behavior in order to stick at all. So, yeah.
Kind of like how our immune system is able to see things that aren't supposed to be there sometimes.
Yeah, so cancer is a perfect example of selfish mutants invading in the population of your cells. Right? So we don't have a really great solution to preventing selfish mutants from invading, although early on, right, we can recognize that there's uncontrolled cell growth, and we have lots of genes that are checks for that. But if there are enough mutations, then there's not a lot we can do about it. But yeah, so your immune system's ability to recognize non-self, I don't know if I would call that a green beard allele, but certainly a good adaptation to have. Okay, so by the way, this is the asexual reproduction cycle. They can also have sex. Okay, so when two of the cells fuse and fertilize, they form a diploid zygote. That zygote can undergo meiosis, right, and you get haploid amoebas again. So here we have a life cycle that's dominated by the haploid stage again, right? The diploid stage is very quick. It's just the zygote and that's it.
So how much about these life cycles do we have to know?
Whatever is on your learning objectives. I don't remember off the top of my head. There are a couple of like, you have to recognize this, the life cycle of this thing and this thing, but it's not the exhaustive list.
But you may not have to, you probably won't, we won't have to like produce them from nothing?
No, yeah. Definitely not. Just a point of clarification from last time. Give me like a 1 if you think that this is an example of alternation of generations, and a 2 if you think that this is not an example of alternation of generations. So yes, this is showing alternation of generations. No, this is not. Okay. So 2's have it. Someone who said 2. Why do you think that, Spencer?
Because it's kind of like an either-or type of thing. They're not guaranteed to go through both within their lifetime.
Okay, that's a good guess, but actually it's much more specific. So there's something that you're looking for to recognize whether you can call this alternation of generations or not. Alex, do you know what I'm talking about?
There's 2 circles and an alternative generator. like circle, you have N and 2N in the same circle?
I wasn't thinking about it that way either. So the way I was thinking about it was, if you have, oh, never mind. That's a terrible example.
Meet the slime mold. For much of the year,
Oh my God. Okay. I had a heart attack for just a minute there. Okay. So, yeah, sorry. Alternation of generations is usually reserved for organisms that are multicellular in their haploid stage and multicellular in their diploid stage. So this is actually not multicellular at all, which is why it's a bad example. But so things like mosses, right, where there's a gametophyte and a sporophyte, and both of those things are multicellular. That's where we get to call it alternation of generations. So it's all about, are there rounds of mitosis as a haploid and as a diploid? So I don't think I was really clear on that last time, but definitely be able to recognize that. Not, well, I mean, it's not multicellular in any stage of its life cycle, technically. I mean, I guess you could make an argument for that, but it's a philosophical argument more than anything. But it's a diploid for exactly one cell, right? And it instantly undergoes meiosis, right? So this zygote doesn't then divide into other 2N cells. No.
And like the other slime mold thing that we were looking at with the feeding Plasmodia and
Nope. Correct.
And then actual asexual reproduction when they're in the group stage.
They don't. Neither of them do. Yeah. Yeah, but what has to be true for alternation of generations? Yes, both of those, the diploid and haploid, have to be multicellular. Okay. All right, so that's it for the slime molds. Again, we've got cellular slime molds and plasmodial slime molds. What's the big difference between those two?
In the aggregate, the cellular keep their identity as a cell.
Yep.
Like they're separated by their cell membranes.
Yep.
But in a Plasmodium, they're just one big supercell.
Yep, with lots of nuclei. Exactly.
Yeah, they don't dissolve the nuclei, but they do get rid of the cell walls.
Exactly, or the cell membranes. All right, next up are tubulinids. So tubulinids, when I think of like the archetypical amoeba, I think of Amoeba Proteus because that's the one we're used to seeing under the microscope, and that's a tubulinid. Okay, so tubulinids tend to have lobe or tube-shaped pseudopodia. Okay, common unicellular protists in soil, common in freshwater, common in marine environments, so they're pretty cosmopolitan. They occur in lots of different places. And they are hungry, hungry hippos.
The predators.
Yeah.
I would love under a microscope to watch one of these hunt. That would be actually really cool to see.
Yeah, so unfortunately, we haven't had a lot of luck finding living samples in lab with this course, but we tried last quarter to look at these under the scope. Okay, the last lineage of Amoeba Zones are the Nth Amoebas. So Nth Amoebas are parasites. So famous example is the Nth Amoeba histolytica, which causes amoebic dysentery in humans, and is a leading cause of death among the protists. Right? Bless you. Okay, that's all you have to know, right? So think Ent, like inside, So these are just amoeba that are parasites. They live inside other things. Okay, there's literally one slide with one sentence on episthocons. Okay, these include the animals, the fungi, and their close protist relatives. Full stop. All we're going to talk about with this chapter. Okay. So clearly, protists, right? You've gotten a sense of how tremendous the diversity is across this group of organisms. So they span everything, right? They're single-celled, multi-celled, heterotrophic, autotrophic, mixotrophic. Just you name it, there's a protist that probably does it. Okay. So they're clearly playing a lot of roles in their ecosystems, but we're only going to talk about two interesting roles that they play, that of the symbiont and that of the producer. Okay. So we've actually talked about some examples of protists playing the role of symbiont. What's one that we talked about today? Lichens, exactly. Yeah. So there's a Clora algae that's a symbiont inside a lichen, and there are dinoflagellates that are called zooxanthellae. You don't have to know that word, but those are dinoflagellates that live inside coral polyps and photosynthesize for them. Okay, so it's basically like free room if you cook. Another really good example is if you look inside the guts of a termite, which we do in 101. Did we do that in 211 last quarter?
I don't think so.
We might have watched it. Oh, was something wrong with it? Okay.
Yeah, something went wrong. There was some issue and we just didn't end up being able to.
Well, so I can, if you guys would like, I can find the video, or I'll send you a link to the video, and you can see like a dissection of termite guts. In your free time, if you want to do that, go for it. And inside termite digestive tracts, right, you find all kinds of bacteria and protists that are breaking down cellulose for termites. Why? Because termites don't make their own cellulase. So they can't break down cellulose, which is unfortunate for them because all they eat is plant material that has cellulose, right? So thankfully, right, they have all these protists that are doing it for them, and once again, it's free room if you cook, right? If you make food for me, I will provide you with a very stable, very pleasant environment with constant wood. Who knew we would describe a termite gut as a pleasant environment? Okay. So I got into a really interesting Facebook discussion with some other instructors at PCC about the word symbiotic. Okay, so you might be used to thinking about this word symbiosis as being a mutualism where both parties are benefiting. Okay, so there are mutualisms that are not symbiotic. For example, flowers and insects pollinating them. Right? It's win-win. Okay, but they're not really intimately linked in terms of their life cycle. Like you don't find one living inside the other one or living really close by it for most of its lifespan, right? It's just the bee visits the flower and that's the end of their interaction, right? But it's a win-win, okay? We just saw two examples of symbiotic mutualisms where it's win-win, right? But one is basically living inside the other organism, okay? So your textbook, and I'm willing to change my model of what I thought of symbiosis as well, also describes parasitism as a symbiosis. Okay, so it's a really close, intimate interaction between two species. It just happens to benefit one and harm the other. Okay, so you can have symbiotic mutualisms, but you can also have symbiotic exploitation. Okay, so good examples of this would be Plasmodium, right, causing malaria. So Pfiesteria is one of the, or that species is one of the species of dinoflagellates that cause the red tides. Okay, and then there are a couple that cause other kinds of diseases.
Doesn't really like if a parasite like causes damage to its host, then I don't understand how that would be symbiotic.
Right. So it's symbiotic because it lives inside the host. So like if you get a tapeworm,
So that's it.
it's in your body, right? If you die, it dies,
So yeah.
but it has probably been quite reproductively successful over its lifespan living inside your intestine. Yeah. So it's probably had a lot of babies that are out there just waiting for a new host. So it's gotten what it wanted from you, right? It has won, right? Even if that individual dies, it was tremendously reproductively successful, which was the whole point, right? In a sense. Okay. But yeah, so there's a lot of theory about like what you should expect if you have like a new pathogen, right, that enters a species. So think like HIV, like pretty new to its interaction with humans. Depending on the ecology of the organism or the parasite, some predictions would be that it would basically evolve to be mutualistic over time, right? For exactly the reasons that you said, like it's in its interest to keep its host alive over a long period of time. But then you see other pathogens that actually increase their reproductive success by making their hosts die quicker, right? So think things like cordyceps. You guys heard about cordyceps?
Oh, but those are the things that go into the ants and like the fungus.
Yeah, yeah. So there's like a species of cordyceps that has a host of like every insect imaginable. So it's not just ants, it's like everything.
Yeah.
And it causes them to change their behavior so that they basically
Present themselves.
yeah,
as a predator.
either they crawl up to like the top of a blade of grass, so they're more likely to get eaten by cows, or they crawl up to the top of a tree so that when the fruiting body like erupts from them, it has a really easy way to disperse to new hosts, right? So it's actually in those parasites' interests to cause their host to die at a certain point, right? So again, depending on the ecology, you can get very different selective pressures. Cholera, right? So cholera is something humans have lived with for a really long time. It makes you really, really, really sick, right? But in the course of doing so, disperses itself really well, right? So cholera, it's a, is it a bacterium? I think so. I'm pretty sure. Someone fact check me. I don't even remember what it is, but it basically it causes you to like have really, really bad diarrhea, right, and just like dehydrate you. So it messes with your ion exchange channels, right? So it basically causes osmosis in your gut, so you dehydrate and you just disperse it through the water column, right?
And so like, I think isn't that like one of the ones that like if you don't have a good way of dispersing your feces?
Yes. Yeah. So John Snow is like the father of modern epidemiology, right? He's the one who like realized that the sewage was flowing towards the well on one side of the street in London, and everybody on that side of the street got sick. No, sorry, not Game of Thrones Jon Snow.
I'm like,
Like,
really?
his name was Jon Snow. And then on the other side of the London street, like the water was flowing away from the well. So like everybody's fecal matter was flowing away from the well source. And like one person got sick on that side of the street. So he was just like, oh, I bet it has to do with waste ending up in the well. And he was right. Okay, so the other sort of ecological niche we're going to talk about are producers. So I think we'll talk about producers and ecosystems a lot more at the end of 213. So you'll get this next quarter. But right, you should know that producers, or primary producers as they're also known, are the linchpin of any ecosystem, right? They are the most important group in any ecosystem. You cannot have a functioning ecosystem without producers, right? Nothing can eat anything if there's nothing that's fixing carbon for you, right? Or nothing that's making organic carbon. Okay, so a lot of protists that we've seen are producers. Somebody give me an example of a protist that's a producer.
I guess those aren't protists.
Not protists, but that's a good example. Huh?
Red algae.
Red algae. Somebody give me another one. Green algae. Somebody give me another one.
Yeah,
Red algae. Somebody give me another one.
That one circozoa.
Gold algae.
That one circozoan. Gold algae. Give me another one.
Stromatophiles.
Which one?
Oh. That's the super group. I don't know.
Yeah, so there are some, right? What else? So diatoms are stromatophiles, right? That are photosynthetic. Plants, yeah. So we have a stromatopile example, the diatoms, and we talked about cercozoans, which are in the R, the rhizarians. Are there any alveolates that are producers? Dinoflagellates, yeah. Okay, yeah. So you find producers in pretty much across the board in all of these protist lineages. Okay, so in terrestrial environments, right, the lion's share of production occurs in plants, right? But in aquatic and marine environments, it's different, right? The protists and prokaryotes like cyanobacteria play a much bigger role as the producers in those ecosystems.
Better any well, I guess you just have to, because like there are no animals or anything like that, right? So it's like it's either protists or plants on that physical.
Or occasionally bacteria. Yeah.
Oh, right.
Yeah. So often the first producers in a new ecosystem on land are lichens, right? Which are protists, or the photosynthetic part are the protists. Okay. So obviously, like nutrient availability is super important in any population, right? But in particular in producers, because they're not limited by light availability. Like they're gonna find light, okay? If they're in the photic zone in the water column, right? But they're limited by nitrogen and phosphorus in particular, right? Some populations are limited by iron, have we talked about algal blooms? I'm sure we talked about it before.
With the red tides, we talked about it.
Okay. But in general, right, if there is a surge of nitrogen and phosphorus in, maybe you got a question? No? Okay. Okay, let's say you spray fertilizer on your farm, okay? So that fertilizer is nitrogen and phosphorus. That's why it helps your plants grow, because your plants are also limited by those nutrients. Okay, but not all those nutrients end up in the soil. If there's rainfall or wind, right, there's runoff from agricultural runoff, right, from your farm. Say that ends up in a sewer system, goes through stormwater, right, ends up in the Columbia River, okay? So now we've got excess nitrogen and phosphorus from your farm that's in the Columbia River, and who can use it? Well, a bunch of aquatic algae and protists, right? And they start photosynthesizing like gangbusters. They reproduce, and you get this giant algal bloom, okay? And it's great for them, right? But when they run out of that nutrients, right, they can't like steal it from other algae, right? So at some point, it becomes unavailable to them. They all die, right? So when the algal bloom stops blooming, when they die, bacteria then start to eat them, right? And bacteria are metabolizing aerobically in the water column, okay? And they're using oxygen.
And then they use them.
Exactly. So they're taking oxygen from the water column to do aerobic cellular respiration, which causes these hypoxic dead zones in the water column, which kills a lot of fish and other marine invertebrates and can like destroy ecosystems. So every summer, that's really dark, right? Every summer, if you look at like satellite images of the Mississippi River Delta, right, you see this huge, like almost like state-sized algal bloom off the coast, right? So these things can be big, and it's because of agricultural runoff all along the Mississippi River. So that all ends up in the Gulf of Mexico. There's a big algal bloom, which causes a hypoxic dead zone after the bacteria are done with the algae, which causes a lot of fish death and is of ecological concern. So here's an example of a food web in the open ocean, right? So you've got prokaryotic producers like cyanobacteria, you've got protist producers, looks like we've got some diatoms, maybe some dinoflagellates here, okay? So they're all photosynthesizing, and then there's a bunch of things that are eating them, right? And then there's a bunch of things that are eating them, right? Even some of these guys.
Wait, so carnivorous plankton can eat the other consumers?
Yes.
For like what, when they die, or just like when they
Not
die?
even. Yeah, so these can be secondary consumers. They can eat other plankton.
Like small scale deer and wolves.
Like small scale what?
Deer and wolves. Like
Yeah, basically mini wolves.
the prokaryotic producers and the participant producers are the grass, the herbivorous plankton are the deer, and
Yep.
the carnivorous are the wolves.
Exactly.
Just on a very, very, very tiny scale.
Okay. So one more environmental slide, then we'll take our 5-minute break and I'll switch chapters. Okay. So one of the big concerns with global climate change is that we're actually warming the surface temperature of the ocean. So global average surface temperature in the ocean has gone up over the past couple of decades. And for physical reasons that I don't fully appreciate, this actually reduces the rate of upwelling, right, of nutrient mixing from lower parts of the water column. So there's just not as many nutrients ending up in the photic zone as there used to be.
Yeah, because when the water would cool, it
Would sink.
sinks.
Right.
And
that would cause.
And that just causes this big
Exactly.
cycle.
Exactly.
But when they're closer in temperature, then it doesn't cycle as much.
Right. So what you're looking at is changes in sea surface temperature, that's the SST, since 1950 across the globe. All right, so you can see there are regions in the oceans that have gotten hotter than other regions. And there's actually a couple of regions, right, in the far latitudes that have gotten a little bit cooler on average. And they actually measured the concentration of chlorophyll in milligrams per meter square per year of all these different regions in the ocean. And almost across the board, chlorophyll content in the sea surface has actually gone down. Okay, so it doesn't look like a lot, right? These are really small numbers. Right? But think about how big the ocean is. Okay, so if there's not that, if there's that much chlorophyll missing, it means there's that much primary productivity missing from those ecosystems, which by definition means those ecosystems have to be smaller, right? So there are fewer individuals in those ecosystems. So even these minor little changes in things like global surface temperature can have big impacts on ecosystems. Okay, but now we know about it, and now we can do something about it, right? So let's take a 5-minute break, and I will see you guys for the new chapter at 6:08.
29, right?
Hmm?
29, part 1.